Thank you for inviting me to be here today. Good morning, everybody. So I'm going to be talking about Gene Matcher, and I don't know if many of you know about it. I know some people here that know about it and use it, but I don't know if most of you know about it. So Gene Matcher, it's a tool that we actually developed as part of the Baylor Hopkins Center for Mendelian Genomics to help us with the analysis of our research whole exome sequencing. And you heard about the Centers for Mendelian Genomics, so I'm not going to tell you more about it. This is the website where you can submit patients to the project. And the goal of the project is to sequence about 1,000 whole exome sequencing per year of patients with rare Mendelian phenotypes to try to identify the molecular base of these phenotypes. So when we started doing that, we started finding sets of genes that were good candidates for each one of the families that we were working on, but then we have to pick one gene out of 5 or out of 10 that is the right one or that could be the good candidate to be causative in these families. How can we do that? Like, there are many ways to us to find support to call a gene causative. So you can look at the frequency of the variant in the general population. You can look at how often this gene is mutated among healthy individuals. You can perform functional studies on this gene and see what pathways are affected by the mutation in this gene. But the main one that we always are looking for is to find at least 2 or 3 or 4 patients with the same phenotype and mutation in the same candidate gene. So this is what actually makes us more certain about this gene being causative for this family. The way to do it when we are working with rare Mendelian phenotypes, it's really complicated, right? Some of these patients, we find how many of them around the world, like 10, 15. So how can we find more patients? The way to do it is to put people to talk to each other. If we are not talking to each other and if we are not sharing the data that we have, we can really not get anywhere nowadays. So we were trying to find it in an efficient way, an easy way that would be fast and that would not require too much effort from the people who are sharing. And this is when we created Gene Matcher. So this is the website. You can go there, create an account, and submit an entry with your gene or genes of interest. And I'll tell you a little bit of how it works and what we have achieved with it as of now. So again, it's intended to find all the individuals that have interest in the same gene that you are studying. And you are studying this gene because you have a patient with mutation in this gene or because you have an animal model with a mutation in this gene, and you think this gene is causative of the phenotype of interest. Because we started by matching on gene, as the name says, gene match is you put a gene of interest in there, and if someone else in the database has a gene of interest that is the same as yours, the database will connect both of you. So because we are working with gene names, there is no identifiable data in the database. So you don't have to worry about IRB and consent forms to put your data there. The matching is automated in a way that as soon as the database identifies 2 people with the interest in the same gene, an email is sent to both at the same time. And I will know that Megan has interest in gene X, and Megan will know that I have interest in gene X, and we have our email contact now, and we can talk to each other. So we have a problem in science, and we think Gene Matcher is helping to change the behavior of scientists a little bit, and researchers and clinicians, that is people is very much afraid of sharing data. They are very much afraid of putting out there what they have, and they think that the other will pick the data, scoop them, and go on and publish it without telling them anything. So when we were trying to deal with our submitters from the Baylor Hopkins Center for Mendelian Genomics, it was a problem. People were always afraid of, like, if you tell them what is the gene, they will just use that to do their publication, scoop us. So Gene Matcher kind of deal with this problem by forming both at the same time. And all I know is that Megan is interested in the same gene as I am, but I don't know anything else, so what can I do with it? So I think it has helped people to be more willing to share the data and to collaborate because they feel more safe. Unfortunately, we have to deal with it, and you still have to deal with it. And then now you also have the choice of choosing who you want to collaborate with. If I see Megan's name and I don't like Megan, I just don't send an email to her. You can choose who you want to exchange more data or not. We do like each other. And then we started later adding the phenotype matching. So you can also now in gene match by features. What is very much more complicated? You can imagine that matching by features sometimes can be very unspecific. If all my patient has is intellectual disability and microcephaly, I'm going to match to a lot of patients out there that are not the same as mine. So, but we have been dealing with it for the last year and trying to make the algorithm better. You can actually put together gene and features, what makes the match more specific. So it's available if you want to try. So right now in Gene Matcher, you can match based on the gene name, genomic location, so it makes a little bit more specific. Like if you want a specific variant out there, you can put the genomic location, try to find a patient with mutation in the same position. You can match based on features, and you can also match based on the OMIM number of the phenotype. Like if you want patients with double VITs, you can get the OMIM phenotype number for double VITs and match with all the users that have interest in the same disease as you. So one thing that's new in Gene Matcher, and maybe people that have been using Gene Matcher still don't know about it, it's like we started with researchers and clinicians using it mostly. And then we started getting patients trying to use Gene Matcher, and we were trying to find a way that would be useful for everybody and not exclude the patients from the database. So one way that we started doing is we now ask people to tell us who they are. Like, are they researchers? Are they healthcare providers? Are they patients? And you can choose who you want to match to. So if I'm a researcher using Gene Matcher and I do not want to match a patient because I do not think I'm able, or I don't have the time to be talking to a patient, because patients will really try to find out about it more, like sometimes more than clinicians and researchers want to. So you can choose. And if a patient is not interested in finding a researcher, but is also only interested in finding a clinician, he also can choose. So you can go to Gene Matcher and you can choose who you want to match to also. We thought that it would make it easier to people to use the database without getting frustrated with a lot of emails that they don't want to receive. Everybody who works on any kind of animal model is welcome to use Gene Matcher. The name of the gene is also based on the human gene name, but you can be a person working on a fruit fly. You can be a person working a mouse model. You have a gene that you cause a phenotype in one of these animal models, and you want to see if there is a human phenotype that matches the animal model that you're working on. So you can tell us what animal model, what species you're working on, and go from there. You can put the gene name in there. And another new thing that we added is people have asked us to have more information on the matching email. So we now added, like, you can tell us if the variant is heterozygous, what kind of variant it is. Is it a stop gain? Is it a missense? Is it a indel? Whatever it is. And you can also tell us what mode of inheritance you are working on in your family. If you give us this information, the information will go in the matching email notification. And then the other person will have an idea of if this is a match that they want to follow up on. Because, like, if you say that your variant is heterozygous and my variant is homozygous, maybe I don't want to follow up on that email with you. Or I can prioritize and follow up in other one that I think will be a better match to my variant and my gene. So this is out there also now. This is how a matching email will look like. So in here, this is my family, and I have the identifier of my family, the name of the gene, and I had added this information about my variant and my gene and mode of inheritance. The other user, now I know who the person is. I have the email contact, and I can contact her and ask more information about the phenotype of the patient. Do you have a patient? What is the phenotype of the patient? What is the variant? She didn't tell me anything about the variant being heterozygous, homozygous. I can now ask her about the mode of inheritance and everything else that she's thinking, and we can go from there and see if this is a match and get more support to call this gene causative. This is all to get more support to prove that the gene is causative. This is what we are looking for. So some of the numbers in GeneMatcher right now, as of October 1st, we have 5,293 genes submitted by 2,259 submitters. We have had 9,920 matches on 1,781 genes. So this is actually very good because it tells us that these genes that are there are good candidates. Not all of them match with everybody all the time. So people are being very selective on putting good candidate genes in GeneMatcher, what makes the matches more productive. So out of the 22,000 genes that we think we have in our genome, 3,633 of them are actually known to cause a phenotype. And so we have 18,000 genes, about 18,000 genes that we don't know what the phenotype they could cause, a mutation in these genes could cause. In GeneMatcher, we have, as I said, 5,293 genes. 1,285 of them are actually genes that have a known phenotype in homine. So people are maybe looking for phenotypic expansion, trying to identify a variant that was a variant of unknown significance and trying to see if they can find support to call it benign or pathogenic. But we have 4,008 possible novel disease genes in GeneMatcher. So out of these 18,000 that we have left that have no phenotype associated to them, 4,000 of them are in GeneMatcher. 4,000 of them, someone out there thought that may cause a phenotype and is looking for more support to call it causative. So it's in GeneMatcher, and if you have a candidate gene, maybe it will be one of these 4,000 and you find a match and you find that it is really causing the phenotype that you are studying. So I think it's very interesting that we have these 4,000 genes in there out of the 18,000. I'm actually very excited about them. I hope they will be proved to be pathogenic or not soon. Another thing is like people have been very worried about, oh, do I get a lot of matches in GeneMatcher? Do I get a lot of unspecific matches or false matches in GeneMatcher? Just to have an idea, out of the 2,259 submitters that we have there, 686 of them never got a match. So really, again, it tells us that the genes that are in GeneMatcher are good candidate genes and that the matches have a good chance of being productive for you. We have 68 countries using GeneMatcher as of now, and I'm not going to talk about the results of the survey that we are now. There's a survey out there. If you didn't take it yet, please do so. We are trying to get more information about what happens to the matches. We do not have control over the matches, so we don't know what I know that I matched to Phil, but I don't know. Oh, I know that Megan matched to Phil, but I don't know what the gene was. I don't know what happened after that. So we are trying to gather information about what's happening to the matches. So this is the intention of the survey. I'm not going to go over that because I'm about to stop. I just want to tell you that GeneMatcher is also part of the Matchmaker Exchange group, and the Matchmaker Exchange group is a group that actually is connecting different databases that do more or less what GeneMatcher does. And the idea is that you do not have to go to each one of these databases to do a query for your patient. You go to one of them, and from there, you send your data to the others. So right now, from GeneMatcher, you can go to Australian Genomics Health Alliance, the Broad, Decipher, MyGene2, and Phenome Central. We have had 324 matches with Phenome Central, 538 matches with Decipher, 122 matches with MyGene2, and 24 to the Broad. I have to say that the Broad and MyGene2 are newer. We have been connected to Decipher and Phenome Central for much longer, so this is why we have more matches to them. These are some of the, in GeneMatcher you have a publication page where you can find the publications that were possible thanks to a match in GeneMatcher. If you have one that is not there, we are happy to put it there, so send us an email, tell us that you got a paper that was based on a match by GeneMatcher, and we are happy to put it there.